Welcome to this explainer. Right at this very second, there's a literal microscopic detective story unfolding deep inside your own body. We're going to be looking at the MHC Class II Antigen Presentation Pathway. And yeah, I know that sounds like a massive mouthful of science jargon, but stick with me, because by the end of this, you are going to be absolutely amazed at the sophisticated surveillance network keeping you alive every single day.
Think about this for a second. How do your immune cells actually see a virus? It's one of those things we kind of just take for granted, right? We just assume our immune system magically knows the moment an invader breaks in. But here's the really surprising problem. The key coordinators of our adaptive immune system, the very cells in charge of organizing the counterattack, are actually totally blind to hidden threats. On their own, they literally can't see what they're looking for.
So, how does the body solve this blindness? It all comes down to a core concept called antigen presentation. Basically, it's a process where an antigen, which is just a tiny fragment of a pathogen, is physically held out and presented to those blind T cells on a specialized molecule called MHC. It's kind of like a frontline scout holding up a crucial piece of evidence for the general to inspect. Without that explicit presentation, the T cells would just drift right past the threat without batting an eye. Okay, let's dive right into this. Here's how we're going to break it down. First, the blind immune system. Second, meeting the sentinels. Third, the 5-step presentation. And finally, triggering the alarm.
All right, part 1, the blind immune system, or why T cells really need our help.
Now, T cells are absolutely incredible. They're the ultimate commanders and killers of the adaptive immune system. But they have some pretty severe limitations. First off, they can't look inside a cell to see if a virus or a bacterium is hiding out in there. And secondly, they can't even recognize free-floating antigens on their own. Imagine a police officer who can't see criminals hiding inside a house and can't even see them running down the street. To actually do their job, T cells absolutely require what we call a cellular billboard, so they can scan for health or infection. And that perfectly sets up the evolutionary solution to this blindness problem, the major histocompatibility complex, or MHC for short. In humans, you'll also hear this called the HLA, or human leukocyte antigen complex. This is a tightly linked cluster of genes that codes for specific cell surface proteins. These proteins act as those essential cellular billboards we just talked about. They display fragments of proteins from either inside or outside the cell, meaning those scanning T cells can finally read the evidence and figure out if a cell is healthy, infected, or even cancerous. Moving right along to part 2, let's meet the sentinels, the true scouts of the immune system. Here's the thing, not just any cell gets to wield this MHC Class II billboard. It is highly exclusive. It's only found on specialized antigen-presenting cells, or APCs. We're talking about your dendritic cells, macrophages, and B cells. So what do they display? They specifically show off exogenous antigens. That means extracellular pathogens or toxins that the APC literally engulfed and digested from the surrounding environment. And who exactly are they talking to? They report directly to helper T cells, specifically the CD4-positive ones. If we could translate what this specific MHC Class II pathway is doing into a single urgent message, it would be this. The sentinel cell is essentially screaming, "Hey, look at what I found outside grazing in the tissue. Let's organize an immune response to go hunt it down." It is a frantic, high-stakes call to arms based on raw evidence pulled straight from the battlefield. Which brings us to part 3. The 5-step presentation, where we actually process the enemy. Let's see how this builds. We've got a really clear, high-level roadmap of the 5 phases here. It kicks off with endocytosis, moves right into digestion, then transport and shielding, loading, and finally, presentation and activation. Starting right off with step 1, endocytosis. Picture our antigen-presenting cell. Let's say it's a macrophage, acting like a scout out on patrol. It stumbles across an extracellular pathogen, maybe some wandering bacteria, and physically engulfs it through a process called phagocytosis. It swallows the invader whole, trapping it inside a specialized internal vesicle called a phagosome. Boom, the enemy is captured, and keep in mind, this exact process is happening in your lymph nodes by the millions as we speak. Immediately, we hit step 2, digestion. The cell isn't just taking prisoners here, it destroys them. That phagosome containing the trapped invader fuses with a lysosome, and lysosomes are packed with highly destructive acid and enzymes. It's basically a microscopic stomach of doom. In a flash, the pathogen is completely shredded into tiny little pieces, which we call peptide fragments. The invader is dead, sure, but its remnants are about to become incredibly useful. Now, what's absolutely fascinating to me is what's happening at the exact same time in another part of the cell for step 3. Over in the endoplasmic reticulum, the fresh MHC Class II molecules are being synthesized. But there's a huge risk here. What if the MHC molecule accidentally grabs a piece of the cell's own healthy proteins and displays that by mistake? To prevent a catastrophic friendly fire incident, the MHC wears this clever, temporary straitjacket called the invariant chain. This shield completely blocks the binding groove, making sure it doesn't accidentally pick up cellular peptides while it travels. It is such an elegant safety mechanism. And that brings us to the critical moment in step 4, loading. The vesicle carrying our safely shielded MHC Class II molecule finally fuses with that late endosome, the one holding the digested, shredded pathogen fragments. That safety shield, the invariant chain, is finally chopped away. And with the groove now exposed, a high-affinity foreign peptide from the digested enemy is firmly loaded right into the MHC molecule. The evidence is completely locked in place. And now for the grand finale, part 4, triggering the alarm, or what I like to call the immune handshake. Step 5 is presentation. That newly loaded MHC Class II complex, proudly holding its tiny piece of the enemy, travels all the way up to the surface of the cell. The cellular billboard is officially raised for the outside world to see. This right here is the climax of the entire pathway. The CD4-positive helper T cell finally arrives and physically docks with the MHC Class II molecule. This is the ultimate handshake between the antigen-presenting cell and the helper T cell. The blind commander has finally been handed the evidence, and it can now clearly see exactly what is attacking the body. And once that connection is made, it is game on. An all-out immune war is triggered. The helper T cell immediately starts pumping out these powerful chemical signals called cytokines. These cytokines cascade through the body, activating B cells to start churning out highly specific antibodies against the invader. Simultaneously, they activate more macrophages, sending them into a total frenzy to hunt down and destroy even more pathogens. This massive, perfectly coordinated defense all stems from that one single vital communication step on the cellular billboard. We've just seen how flawlessly this system captures, processes, and displays threats. But I want to leave you with a pretty provocative thought. If our cells have to constantly put these protein fragments on display just for us to survive, what on earth happens when an evolving pathogen figures out how to hide from the billboard altogether? It's an intense, microscopic arms race, and it never truly ends. Thank you so much for joining me on this explainer, and definitely keep questioning the incredible machinery inside you.